Hi, my name is April Sagan. I am a postdoc at the University of Pittsburgh and the UPMC Hillman Cancer Center. The work I'm going to present is on modeling gene regulation using spatial transcriptomics data. I'll talk about the challenges of spatial transcriptomics and the unique insights that having spatial information can give us. Spatial transcriptomics works in a very similar way to single-cell RNA-seq, except for instead of using a droplet-based method and isolating cells and assigning barcodes that way, barcodes are assigned in a way that preserves the spatial XY coordinates of each transcript that we sequence. The data I'll demonstrate comes from 10x Genomics using their Visium platform. The samples are from a HER2-positive, ER-positive breast cancer tumor, and the H&E stained microscopy image is shown here in the bottom left. We can overlay on this image the expression of the genes CRISP3 and CXL14, for example. What makes this unique from single-cell RNA-seq is now we have where in the sample these genes are expressed and the spatial patterns that they follow. One important caveat is that these little spots, these circles here that are highlighted according to gene expression, actually can contain upwards of 10 cells. And so we need to develop tools that can account for the fact that we're measuring the sum of gene expression of multiple cells and of multiple cell types. We're modeling gene expression profiles in each spot in terms of transcription factors, which are proteins that regulate gene transcription. We're using prior knowledge about TF gene interactions to calculate a biologically meaningful form of dimensionality reduction, with the goal of being able to recapitulate the expression of thousands of genes using weights from only a few hundred transcription factors. So in this example here, if I see that gene 1 and gene 2 are both upregulated, but gene 3 is downregulated, I would look at what these have in common, which is transcription factor 1, and I would say that this would get assigned a higher weight in our model than transcription factor 2. For the breast cancer data we're using, we use a previously developed tool to do cell type deconvolution, which estimates the proportion of each cell type in a spot. Here I'm showing the proportion of T cells and epithelial cells, along with a marker gene for each. The next figure shows the inferred transcription factor activity from our model for a handful of TFs. To test that our model is giving sensible results, we see FOXP3 and B-core both being correlated with T cell proportions, which makes sense because there's a good deal of evidence that suggests these two TFs play an important role in T cell regulation in breast tissue. Likewise, GATA3 and ESR1 are important transcriptional regulators in breast epithelial cells. In each spot, we have an upwards of 10 cells, and they're often not all the same cell type. So to calculate the average activity of a TF for a cell type, we model TF activity in a spot as the sum of the TF activity for each cell in that spot, and use a linear regression model with the cell type proportions as feature variables to predict transcription factor activity. The dot plot shows the coefficients from the model, which tells us the activity of each transcription factor in each cell type. For the transcription factors shown here, the model fits the data very well, with an R-squared value higher than 0.8. This tells us that the TFs are expressed consistently in all cells of a given cell type throughout the sample. So, for example, we see FOXA1 and ESR1 in this list, and we see that they are both highly expressed in epithelial cells, and that expression depends mostly just on the cell types in the spot. When the model doesn't fit the data very well, that tells us there's something else that's driving the variance in transcription factor activity. To better model these TFs, we developed a clustered regression model that finds regions where TF activity is more consistent within cells of the same cell type. It works by simultaneously fitting multiple linear models to the sample and then clustering the spots based off which sample fits best. Here I'm showing the results from the clustered regression model for two transcription factors, RIRG and ELF1. For RIRG, we see the methods very clearly separates the sample into two regions. And in each of these two regions, the model fits the data pretty well. The dot plot shown in the lower left-hand corner shows the cell type specific RIRG activity and that it differs between region 0 and region 1 as defined in these two panels. This tells us that the effects of RARG depend not only on the cell type proportions in a spot, but also some things about the spatial context of the spot. And this is the result that we expected to see because RARG is actually a ligand-dependent transcriptional regulator. To summarize, we created a methodology for inferring transcription factor activity from spatial transcriptomics data, which we used to find cell-type-specific transcriptional programs and identify transcription factors whose regulatory effects depend heavily on spatial context and differ within a cell type. To learn more about our lab's work, you can go to osmondbioglulab.com. And thank you for listening to my presentation.